Morning. My topic is coagulation of blood. Coagulation or clotting of blood. It is the conversion of fluid part of blood to a thick jelly known as clot or thrombus. Normally, clotting takes place when blood sheds out from injuries or wounds. So the solid clot seals up the broken tip of blood vessels and prevent further bleeding and loss of blood. Thus, coagulation of blood is a major chemical defense against the loss of blood through injuries or wounds. And the yellowish or straw colored liquid oozes from this clot. It is known as the serum. It is simply blood plasma without these clotting factors or clotting proteins. And about the clotting factors, there are two types: plasma coagulation factors of plasma clotting factors, and four platelet coagulation factors, and a tissue coagulation factor which is a member of plasma factors. And blood coagulation is net result of complex series of enzymatic reactions. First of all, first reaction is the formation of the active enzyme thromboplastin from inactive proenzyme thromboplastinogen with the help of activating factors. And the next one, the second one, is the conversion of the plasma protein prothrombin or the thrombogen into active enzyme thrombin with the help of thromboplastin and several plasma coagulation factors, and also calcium ions. And the third one is the conversion of soluble plasma protein fibrinogen into soluble fibrin with the help of thrombin. Fibrin forms the thread of clot and it forms loose lacework in which blood corpuscles get entangled, forming the finer clot. Next is the enzyme cascade theory. Blood coagulation is the net result of complex series of enzyme catalyzed biochemical reactions, which we call as the enzyme cascade system. Here, the enzyme cascade mechanism involves two distinct routes, that is, the extrinsic pathway and intrinsic pathway. In the extrinsic pathway, thromboplastinogen is released from the damaged tissue cells, but in the later it is released by the breakdown of blood platelets. So these two pathways later on merge together. First of all, we see about a extrinsic pathway. The extrinsic pathway of blood coagulation begins with begins with the release of lipoprotein, extrinsic thromboplastinogen, from the ruptured part of the blood vessels from the damaged tissues around, and this thromboplastinogen reacts with the various clotting factors such as 4th, 5th, and 7th, and also 10th, and gets transformed into active thromboplastin or the extrinsic thromboplastin. This conversion this conversion of plasma proenzyme this conversion represents the first stage. So this is the first stage. Here, thromboplastinogen reacts with the clotting factors 4, 5, 7, and 10 and gets transformed to active thromboplastin, or it is also called the extrinsic thromboplastin. So this is the first stage. The second stage of extrinsic pathway involves the conversion of plasma proenzyme prothrombin into active enzyme thrombin. So here, plasma proenzyme prothrombin, it is converted into active enzyme thrombin. Then the prothrombin is produced and raised by the liver. So it reacts with the extrinsic thromboplastin and the plasma clotting factors 4, 5, 7, and 10. They are transformed to active thromboplastin. And then the third stage, plasma protein fibrinogen. Here, the plasma protein fibrinogen get transformed to insoluble fibrin by reacting with thrombin and the plasma clotting factors 4 and 8. So in the third stage, what happens is that the plasma protein fibrinogen gets transformed to insoluble fibrin by reacting with thrombin and the plasma clotting factors 4 and 8. And initially the fibrin is a loose meshwork of interacting strands, and it is converted into tight and dense aggregate by the formation of covalent cross links between these strands. So this it is converted into tight and dense aggregate by forming covalent cross links between them itself, forming the strands. So in between the strands there are covalent cross links. So it forms a network, dense aggregated network. This reaction is catalyzed by the clotting factors 4 and 8. An initial fibrin network, this forms this intact fibrin network. So the fibrin network formed by the crosslinks between the strands forms the clot. So in the measures of the fibrin network, the blood corpuscles of the blood cells get entangled and forming a jelly like plug. This plug forms the final clot. This plug seals up the broken blood vessels and prevent further loss of blood. So no blood oozes out after the formation of the clot. So from this clot, clear yellow liquid oozes out, that we have said before. This yellow fluid which oozes out, it is called the serum. And the next is intrinsic pathway. This intrinsic pathway of blood clotting, it begins with the adherence of numerous blood platelets to the ruptured areas of blood vessels. So numerous blood platelets gather together at the ruptured part of the blood vessels. This dumping or the accumulation of blood platelets cause the disintegration, releasing platelet clotting factors to blood plasma. So to the blood plasma, platelet clotting factors are being released. So if it is a small injury, the platelet dumping seals up the injury without initiating clotting reactions. There will be no reactions, but it soon seals up the injury. The first stage of the intrinsic pathway involves the conversion of inactive intrinsic thromboplastinogen to active intrinsic thromboplastic. Here, in this case, the thromboplastinogen react with plasma clotting factors. Here, the plasma clotting factors in which the thromboplastinogen reacts are the 4th, 5th, 8th, 9th, 10th and 11th and also 12th. And the platelet clotting factors. So, there are plasma clotting factors and platelet clotting factors. Platelet clotting factors are PF1, PF2, PF3 and PF4. So, in the second stage, prothrombin gets converted into thrombin. So in second stage here, prothrombin get converted to thrombin by reacting with intrinsic thromboplastin, and the plasma clotting factors. To intrinsic thromboplastin and plasma clotting factors 4, 5 and 10. And the third stage, fibrinogen gets converted to fibrin by the reacting with thrombin, the plasma factors 4 and 8 and the platelet factor PF2. Here, so in the third stage, the platelet clotting factor taking place is the PF2. So initially fibrin forms a loose meshwork over the injured part. So it is zone stabilized to a dense network by action of plasma factors 4 and 8 as in the extrinsic pathway. Apart from converting fibrinogen to fibrin, thrombin stimulates the dumping and also the accumulation or dumping, accumulation and also the disintegration of more and more platelets, releasing more platelet factors. Thus the clot is formed. So this is all. Thank you.